Good afternoon, everyone. How are you? Everyone's doing well. My name is Scott Peters. I'm the representative for San Diego and La Jolla, where our guest comes from. We are one of the centers of biomedical research, and I'm a big advocate of strong funding for basic scientific research, not just for my district where it's very important, but so we can keep our lead as a nation and continue to make discoveries and inventions and cures and businesses right here on our shores. Pleased to welcome and introduce Dr. Larry Goldstein. Larry's a leader in human embryonic stem cell research, distinguished professor at University of California, San Diego, Department of Cellular and Molecular Medicine, and the Department of Neurosciences. He's also the principal investigator at the UCSD Goldstein Lab. Larry received his undergraduate degree at UC San Diego and further pursued education at the University of Washington in Seattle, University of Colorado, and MIT, and was on the faculty of Harvard University from 1983 to 1993 as a tenured professor. He returned to UCSD in 1993 as a member of the faculty. In November 2013, he was named director of the Stanford Stem Cell Clinical Center, and his research is focused specifically on Lou Gehrig's disease, Alzheimer's disease, and others. His lab has successfully recreated neurons from stem cells that show the effects of Alzheimer's. Dr. Goldstein is a member of the American Academy of Arts and Sciences, the American Society for Cell Biology, the Sanford Consortium for Regenerative Medicine, and was previously a member of the Cell and Developmental Biology Department at Harvard, and recently retired from the Howard Hughes Medical Institute. He's a strong and influential leader in the UC School of Medicine, and Dr. Goldstein continues to work tirelessly to find cures for the deadliest diseases in and out of the lab. He's testified before both chambers of the United States Congress. I hope they were listening to you, Larry, regarding stem cell research and at the National Institutes of Health. It's a real privilege and an honor, and I'm very proud to introduce Larry Goldstein. Thank you.
All right, good afternoon. I hope you all can hear me. I'll struggle a little bit with my voice, but we'll get through it, I hope. So first of all, I want to thank Congressman Peters. He's been a real forceful advocate for science in my district, and as a scientist, I can tell you he gets it. The American investment in science and technology is what will maintain our national competitiveness moving forward, not just in medical science, which I care most about, but in all areas of science, in particular those in San Diego, which is a leader. And of course, I am sufficiently nationalistic to care about my home state and county, where Congressman Peters' efforts are greatly appreciated. Sorry about my voice, hang on a sec. All right, so I want to leave you at the end of this with two take-home messages. Take-home message number one, the American investment in science, when you look at the rate of return over 10 to 20 years, pays us back at the rate of 20 to 25% annually is our return on investment. It's a fantastic, valuable thing that we do in science in this country. It's not the only reason to do science, but if you only look at things through the economic lens, the numbers work. They don't work for private companies, but they work for the country. That's take-home message number one. Take-home message number two is that stem cells are an increasingly important part of that national investment annually. And my job today is to tell you about recent progress we and others have made using human stem cells of all sorts to conquer some of our deadliest diseases. And so my topic, although initially focused on embryonic stem cell research, I'm going to broaden and talk briefly about each of the different areas of stem cells, from adult through reprogrammed to embryonic, and try to give you a flavor for how each is helping us solve problems in medicine. Now, I'm actually going to go back 15 years to testimony I gave along with Christopher Reeve in the Senate Appropriations Committee. And Chris, you may recall, was a victim of a spinal cord injury, which regrettably shortened his life. But Chris was an indomitable spirit, and in addition to starting the Christopher Reeve Paralysis Foundation, he had an abiding belief that medical science, in particular medical science with embryonic stem cells, would someday cure him and people like him of the devastating effects of spinal cord injury. And I'll tell you at the end about 2 projects that we think will do exactly that in the coming years. There really has been progress on this front. But of course, the initial starting point is, what the heck is a stem cell, and what will they be able to do to help us solve problems in science and disease? And so I want to just give you a very brief definition of a stem cell. So how many of you took high school biology? All right, good. And how many of you learned the following thing? Cells grow by dividing, y'all learned that, and you learned that the 2 daughter cells from a cell division were identical. Does that sound familiar? Raise your hand if that's what you learned. All right, that's what we call the biggest lie in biology. So why do I say that? What do I mean by that? Well, if you look at me, you'll see that I have skin and eyes and hair and heart and lung. All those cells are arguably and obviously different. They have different functions, they look different, they do different things, and they turn on and off different parts of the DNA to make products. And so if that simple high school biology conception were correct, you look at me, I can't possibly be a conglomeration of identical cells. Now, the answer to that conundrum is complex, but part of the answer is in the nature of a stem cell. Because a stem cell is a cell that when it divides, the 2 daughter cells are different. One daughter cell remains a stem cell, and the other daughter cell goes off to adopt a specialized job. I do want to just take a brief moment here. A very distinguished friend of science has just walked into the room, Congressman Rush Holt. I know the congressman will be leaving the House. It's a terrible loss. We need as many scientists as possible in the American government, in my view, and you've done us a great service by taking on this, I know, sometimes thankless job, but from me and my colleagues, thank you, Congressman. We really appreciate what you've done for American science. Would you like, please?
Just a moment, since you were already interrupted your presentation here. Great to see you. Great to see you. I just wanted to remind you all of the reason for this biomedical caucus, and I hope you will take this back to your offices. It is quite simply that we need to continue without politicization, funding for NIH research. I mean, that's pure and simple what this is about. This is investment in the clearest sense of the word. Not that, just as investment, not that every research project pays off, but overall it pays off big for the US government, for the US people. And it is, you know, in recent years, the actual funding, the effective funding for biomedical research has not kept up. Has not kept up with inflation, has not kept up with the need for advancement to push back disease and to increase the quality of life and well-being of Americans. And it hasn't kept up with the opportunity. I don't know the latest statistics for NIH unfunded, excellently reviewed projects, but there's much excellent research going begging. So that's pure and simple why we have this. And we have today another excellent example of why this is important. And And also, I should point out why it might be politicized. And we have to be, this is the sort of thing where we can't let ideology get in the way of human welfare. So that's it. That's why I'm here. That's why you're here.
That's exactly right.
Very much.
Thank you very much.
Great to see you.
Great to see you. Yes,
great to see you.
Yes, thank you to a stalwart leader in this fight. I'll just echo, make no mistake about it, we are in a fight against deadly disease, and we spend far more on disease than we do on research. And I'll come back to some of those numbers in a moment, and you'll get a good example of that. So, maybe the way to return to the subject at hand, having just defined stem cells in the driest, most scientifically precise way. Let's go back to the 60s. Many of you probably saw a film with Dustin Hoffman in it called The Graduate. And Dustin Hoffman received a lot of advice in that movie, some of it about his relationships with older women, which is what I don't want to talk about. But he received a certain amount of career advice, and he was advised, you may recall, that he should go into what? Everybody knows this, plastics. It's incredible. So in 1960, plastics was fantastic career advice because at the time, we built most implements in modern society then out of wood and metal and natural things that we found. And it was the beginning of a revolution that we're now living with, where basically every device you pick up in this room, computers, phones, this remote control, the bottle, all made of plastics. And so in the 60s, plastics was incredible career advice. In 2014, the advice that I would give to the young Dustin Hoffman would be biological plastics, stem cells, because that's what stem cells ultimately are. These are cells that are effectively the raw material of the human body. Whatever the type we're talking about, they are raw material that we can make different parts of human organs and different tissues out of. And as we move forward into the coming decades, these are the biological plastics from which we will build artificial organs, from which we will develop new drugs and devices, and that we will use to understand in a disease we couldn't previously use. So biological plastics is equivalent in a way to stem cells, and that would be the career advice that I would give all of you who are less than 30 years old. Some of you I see are in that category. Actually, some of us in my category have been switching careers multiple times in the American scientific system, which is also a very good thing that we're able to do. So that's the dry definition, cells that can give rise to more of themselves, as well as to specialized daughters that can become heart, brain, kidney, lung, what have you. And of course, as I mentioned, they're promising because they are potentially the raw material to make cells that are lost in disease. Think about type 1 diabetes, juvenile diabetes, where the cells in the pancreas that make insulin are dead and gone. But now clinical trials are beginning to start up with hybrid devices of embryonic stem cells that can make pancreatic cells encased in permeable bags with a local company in San Diego called ViaCyte. And these will shortly go into clinical trials for type 1 diabetics for the first time ever. It's a remarkable technology, but it's a hybrid, creative use of embryonic stem cells and devices made in the bioengineering community that make the cells safe because they're encapsulated. So that's a very important potential use, and there are many such examples, and I'll give you some more in a few minutes. But also, stem cells give us tools to understand and treat disease. So what do I mean by that? So if you have Alzheimer's disease, which I hope nobody in this room does, but if you're one of millions of Americans who do, this is a disease that ultimately kills your ability to think and remember. And you might ask, why don't we have any effective drugs? And the answer, in part, is because we've had to study for years animal versions of the disease. And as you'll see when I come back to this in a moment, animals don't get true Alzheimer's disease. Actually, most animals don't get true human versions of any disease. They are so-called models. And part of our problem, you may know, is that something like 9 out of 10 drugs that enter clinical trials, 9 out of 10 fail. So a 90% failure rate, in part because we don't have human versions of disease that we can use in the lab. I'm going to argue that if we can use stem cells to change the failure rate from 9 out of 10 to 8 out of 10, just going from 90% failure to 80% failure, we will completely, as I suspect, revolutionize this industry. So that's a very important use, is using cells in a dish, as you'll see, to understand disease and to use those cells to test for better drugs that work on human brain cells or other cells, not just mouse or rat or some animal. And so in the sense that I just described, stem cells in a way are what we call enabling technology in the techno community. It's not a one-trick pony. We don't use them for one simple application. They're like recombinant DNA. When I was a young student in the 70s, recombinant DNA was a brand new technology with obvious promise. It was threatening to people because there were concerns that deadly bacteria that had recombinant genes from human viruses would infect the world and get out of the lab. Those concerns turned out to be largely unfounded. But in the interim, we developed a multi-billion-dollar industry in the United States that makes human insulin as we originally expected, makes drugs that keep me alive and many others in our families, and in fact, actually even found their way into the court of law, where recombinant DNA technology is frequently used to figure out who was touching a weapon or whose blood was at the crime scene or what have you. My brothers who are litigators use this technology all the time. And it's an important example because enabling technology is one where you can't necessarily predict what all the uses will be. What you want to do is get those technologies in the hands of as many creative scientists as possible. And if you're me, you want to get the heck out of their way so they can make interesting discoveries. And that's what's beginning to happen now with human stem cells, as I'll tell you. Okay, so in 2014, there are a number of what you would think of as different sources of stem cells. If you only read the accounts in the media, it's very simple. There are adult stem cells, embryonic stem cells, and reprogrammed stem cells. That's it. That's what you get if you read most newspapers or listen to most news channels. Left or right, these are nonpartisan issues. What you are as a stem cell doesn't care whether you're a Republican or a Democrat. In fact, adult stem cells come in dozens, probably, of different types. And in fact, they're actually misnamed. They really ought to be thought of as tissue stem cells. And what I mean by that is, if you think about your skin, you probably know that every day you shed lots of skin. It's the major source of dust in your household, unless you have dogs, which I do, in which case you also have mud and dog hair. But if you're a human living alone, most of the dust comes from you shedding skin. Now, if you think about it, if you just keep shedding skin every day, you are ultimately going to have a problem, right? All your blood's going to leak out. That's bad. And the reason that doesn't happen is that there are so-called adult stem cells in your skin that make new skin every day. That's their job. Now, ordinarily, the stem cells in your skin, so-called adult stem cells, only make skin. In fact, they are strongly restricted biologically to only make skin. And if you think about it, think about the next example. Suppose that I'm a brain stem cell, or I'm thinking about a brain stem cell, my brain stem cells, and instead of waking up today and making neurons, brain cells, they decide to make muscle. That's actually a bad idea. You want stem cells in adult settings to generally be very tightly restricted in what they can do. And that's actually biologically what we observe. You also want their ability to grow to be very limited. In fact, stem cells from most adult sources that we're aware of don't grow very well in the lab, with a couple of exceptions that we'll come to. And again, that's a very important restriction. You don't want your stem cells growing out of control in your body, because when that happens, that's cancer. That's a disease, and that's bad. So you want cells to ordinarily be under very tight control, so they only divide when the system says divide now. And that's what most stem cells do in their daily lives. And that's particularly true of stem cells from adult tissues, such as skin or brain or heart or liver, what have you. We're not sure about whether all organs have adult stem cells in them or not. By the way, there are so-called cord blood stem cells. Those are sometimes mistakenly referred to as embryonic, but they're not. They are actually in the class of cells that we would classify as adult stem cell or tissue stem cell. The most dominant type of stem cell in cord blood is a so-called hematopoietic, terrible word, blood-forming stem cell. It's like the stem cells in your bone marrow, whose daily job is to make new blood and new cells of the immune system to protect you from infection. And cord blood just happens to be a very rich source of those blood-forming stem cells. And finally, there are so-called amniotic fluid stem cells, again, mistakenly called embryonic. Again, they're not. They're probably another terrible word. They're probably some sort of mesenchymal stem cell, but even that is now under question scientifically. None of these sources of stem cells from adults that we know of, when they've been carefully and rigorously tested, none of them is what we would call pluripotent, that is, have the ability to make all of the different adult cell types, unless we genetic engineer them heavily, which I'll come to in a moment. Now then, I want to emphasize, I am not saying that adult stem cells are not useful. In fact, they're incredibly useful. We use them routinely in research on cancer and blood diseases and other diseases of adult organ systems. They're a valuable tool or set of tools in the fight against human disease. But please remember that when you're going to fix your automobile, you need to have a toolbox. You don't want to be trying to turn a bolt with a screwdriver. You have to use the right tool for the right job, and the same applies in stem cell biology. There are terrific opportunities where adult stem cells are part of the solution to the medical or biological problem, and then there are situations where we really don't have good sources of adult stem cells that will let us solve the problem that we're trying to defeat. And that takes us to so-called embryonic stem cells, where there are really two issues. One issue is the ethical issue, and I'm not going to downplay this. Embryonic stem cells come from embryos that are left over after in vitro fertilization treatment of couples who can't have kids the old-fashioned way. If a couple decides to discard those embryos, as some do, cells can be extracted from them, killing the embryo in the process. But those cells, coming from an embryo that would otherwise be destroyed, are incredibly useful in medical research, as you'll see in a moment, because they have the ability to grow more or less indefinitely. I'll just tell you that we use in my lab some of the original stem cells that Jamie Thomson first isolated in 1998. They've been in and out of the freezer, and they continue to grow well and behave themselves by our standards of use in the lab. And they retain the ability to make brain cells, heart cells, blood cells perhaps, all sorts of cells in the adult. Very, very useful cell type, as you'll see again momentarily. But I cannot make the ethical issue go away. There are some people who object to it. There are some people, such as myself, who do not object to it as long as it's well regulated. But the scientific promise of this area of technology is unavoidable. It is quite useful, as you'll see. Now, the final type of stem cell that you may have heard of, because this won the Nobel Prize a couple of years ago for Shinya Yamanaka and John Gurdon, is a so-called reprogrammed stem cell. So what is that? So I'm going to pick on Congressman Holt since he's still in the room. If I took a sample of Congressman Holt's skin So now I've got some of his skin cells. I could treat them with a genetic bag of tricks that Shinya Yamanaka discovered. And I could turn those skin cells in my lab into a type of stem cell called a reprogrammed stem cell. Those reprogrammed cells have many, but not all, of the properties of embryonic stem cells. But they have a very important use, which is I could, pardon the intrusion on your personal life, Congressman, I could make Congressman Holt's brain in a dish. Well, I could make bits and pieces of it. So I could make brain cells in a dish that had his unique DNA and made him the brilliant man that he is, and I could study how his brain cells behaved when I challenged them with different sorts of disease-causing agents or what have you. And as you'll see in a moment, that's been fantastically useful with Craig Venter, where we've actually been doing this. Now, make no mistake about it, all of these types of stem cells are incredibly useful. It's a question of the right tool for the right job. In the area of neurodegeneration, which is the area I work on, Alzheimer's disease and ALS, I don't have a usable source of adult stem cells that I think is going to help me crack these problems. I rely on reprogrammed stem cells and embryonic stem cells to help me fight these diseases, as you'll see. Now, I just want to make one final pitch for you to remember. Every major American city, as well as cities around the world, as near as I can tell, have so-called stem cell clinics springing up that claim to be able to grow stem cells out of fat left over after liposuction. And the claim is that these, quote, stem cells can be used to treat any disease that you may have for $40,000, $50,000, $100,000, depending on the clinic. And I'll just tell you that for the most part, not only are these cells not stem cells, but we're really not even sure what they are. Their isolation methods vary from clinic to clinic. It's a completely untestable set of claims. They don't follow their patients, so we don't know if it ever works. But they're very good at taking your money and shooting something they grew out of fat back into your blood. That's the simple fact. It's sometimes referred to as 21st century snake oil, as 60 Minutes put it a few years ago. And it's a real problem in public health and regulation because these clinics operate in the gray zone of FDA regulation and medical practice. It's a real problem, and people are basically having money stolen from them by many of these clinics. Okay, so current medical needs where stem cells might help. Well, it's the usual list of pretty much every disease that afflicts us. And that's because just about every disease that we deal with on a daily basis is a disease where cells have broken down in their normal functions, such as heart failure or Alzheimer's disease, or have frankly died and are no longer there, such as type 1 diabetes or the late stages of Alzheimer's disease. And so in thinking about diseases where better understanding using human stem cells or the ability to replace cells that are damaged or lost, the list is pretty much everything that you know of, that your families are fighting with. Adult stem cells have been fantastically useful in understanding and treating cancer. Embryonic stem cells are making impact in the area of vision. And as I'll show you, both reprogrammed and embryonic stem cells are helping us deal with neurodegenerative disease and injury. So let me give you an example where reprogrammed stem cells have been very helpful to us. And this is what we sometimes refer to as human disease in a dish. And the examples I'll give you are the ability to use these so-called reprogrammed stem cells, where we take skin cells called fibroblasts and convert them into stem cells that then let us make different types of brain cells, so-called neurons and astrocytes, as you'll see, so that we can study their interactions and test drugs. So one such example comes from the problem of Alzheimer's disease, which is a devastating disease in this country and others. I'll just give you a couple of numbers. So first of all, the disease is common. 10% of us over the age of 65 have Alzheimer's disease. 50% of us over the age of 85. I'm 58, so probably about 1% of people my age have it, and my risk is going to keep on increasing every year. Not only is the disease common, but it is economically crippling to most families that have to deal with it. Currently, the United States spends somewhere in the range of $200 to $400 billion a year taking care of people with Alzheimer's disease. But we spend at the NIH only about $500 million a year, and that's not funding all of the best ideas that might be used to make an impact on this disease. Now, to give you an example of how bad that ratio is, I don't know about you guys, but I don't think very well about millions versus billions and the relative orders of magnitude. If you look at 500 million against a $200 billion cost, you're about, probably about 1 to 400 or so. What that means is the following. Imagine that you have a leaking roof in your house, and you're spending $400 a year to fix the damage from that leak. But you're only spending $1 to find out where the leak is every year. $400 out the door, $1 spent trying to fix it. The ratio doesn't make sense. Surely we can do better than that. And that is ultimately the problem we face with diseases like Alzheimer's. And it's why investment at the NIH is ultimately so important, to try to figure out what the heck is going wrong there. Now, my lab currently works on this problem, and we're trying to figure out what causes the various parts of the pathology, and most importantly, what causes the biochemical changes that happen in brain cells at the earliest stage of disease. And one of the things we've been doing is to take skin cells from patients with rare hereditary forms of Alzheimer's disease, use reprogramming technology to make stem cells that carry the genetic changes that cause hereditary Alzheimer's disease, and then we, amazingly, make neurons in a dish from those same stem cells, and we can see what goes wrong in the early biochemical changes caused by those genetic lesions. Now, we currently have a project in my lab looking for already approved, FDA-approved drugs that fix some of that altered biochemistry in those stem cell-based neurons. It's a project that, given the funding limits at the NIH, would never have been funded at the NIH, but is being funded because I live in California, where we're fortunate to have a bond initiative that is supporting stem cell research. And as a result of state support of that project, we now have literally dozens of candidate FDA-approved drugs that might change the abnormal biochemistry in these Alzheimer neurons and improve it. Now, we know the odds. The odds are we're going to fail with every one of those. On the other hand, it's several dozen leads we wouldn't otherwise have found, except for the ability to use this technology with now state financing because of federal financing limits for a drug discovery project for this terrible, costly disease. Now, it's not that I'm saying we should fund my lab more. No, we should fund dozens of labs doing the kinds of things we're doing, each of which is bringing different ideas about how to use stem cells to understand and treat Alzheimer's disease. But it's one example of how this technology is turning out to be incredibly useful. Here's another example, the infamous, what I call Craig Venter project in my lab. So Craig, as you know, is not only a very vocal American scientist, he's really a brilliant guy, he's also a friend and colleague. And Craig, some years ago, determined his individual DNA sequence and deposited it in public databases, and it's a very high quality DNA sequence. We now know all of Craig's so-called risk variants for different kinds of diseases, including Alzheimer's disease, where he, like many of us in this room, have common genetic variants that increase his risk of disease in some cases or decrease his risk in others. And the question is, how the heck do you solve a problem where each of our individual genetic variation contributes to risk of disease? And so what we've done is we've taken skin cells from Craig, my graduate student, hunted him down, Craig in his Maserati, my graduate student on his motor scooter, and got Craig to give us a skin biopsy, which we then reprogrammed into functional reprogrammed stem cells. And so we really now do make Craig's brain cells in a dish, and we've been studying his genetic variants as well as the same genetic variants in different individual humans. And we've actually discovered how one of these risk factors works, we think, and it's going to have an impact on how we test different classes of drugs in clinical trials. Because it looks as though, we think, that Craig's risk variant, which a third of us in this room have, will change our response to important subclasses of therapeutic agents that are being developed. And therefore, you don't want to test people who have those variants in clinical trials because we would predict they will not respond to those agents. Now, we're not sure of this. This is what we call bleeding edge science. But it's another example of how important this, oh yes, I had a little diagram. There's our smiling inventor there, and there's his stem cells caught in the act. It's an important example of how this kind of technology can reshape what we're doing. Okay, let me give you another example. It comes from Lou Gehrig's disease, ALS. This is a disease where the neurons in your spinal cord that control your voluntary muscles, such as your ability to move your fingers or to draw breath, to breathe, or swallow, those so-called motor neurons die. And we don't know why they die. And we don't, at the moment, think that we have a technology that will let us replace them because of what we call the wiring problem. The motor neurons that control breathing muscles in my diaphragm have their initial connections in the spinal cord, and then they have to wire around to their target in the diaphragm. It's a long distance, and they have to wire properly. And so at the moment, we're pessimistic about solving that problem in the short term. However, as a result of work we've done at UC San Diego, in my lab and many of my colleagues' labs, as well as some competitors at Harvard, have to mention them, we now know that the cells that are dying are not necessarily the cells that are entirely at fault. And so we're working on a project now to replace or to add to cells in the neighborhood of the dying motor neurons. We're trying to replace or use so-called astrocytes, which are one of the support cells. Part of their job is what we euphemistically call taking out the trash, is what they do as part of their day job, in order to keep motor neurons from dying in the first place. Now, if this works, it'll be the first effective therapy for ALS, because currently we basically have nothing. And of course, if you're diagnosed with this disease, your average lifespan is predicted to be 3 to 5 years, with some people dying within a year. It's a virtual death sentence, and we got nothing right at the moment. And so what we and others are working to develop is to use either human embryonic stem cells, or, as has been done in a trial at Emory, so-called fetal spinal cord stem cells, and then transplanting those into spinal cords of patients with ALS so that these cells will generate a new, fresh batch of astrocytes in the spinal cord. And based on our work in animals, which we now want to move into humans, the prediction is that these cells will protect motor neurons from continuing to die. So we're now moving towards clinical trials, we hope, in the next few years with human embryonic stem cell versions of this therapy. A company called Neuralstem has been developing a fetal version of this therapy, and there are other different versions of this being tried around the world. And that's the deal about experimental medicine. You have to try a number of different approaches to figure out what's going to work. But we're optimistic that we will be at least partially successful here. Let me close with the final example, which is the one that got Chris Reeve going originally, which is spinal cord injury, where, of course, as a result of a break or compression in the spinal cord, signals can't travel down the cord from the brain out to hands or to breathing muscles or to legs, depending on where the injury occurs. Now, it turns out that if you're a rat with a spinal cord injury, we have a number of different approaches with embryonic stem cells or with fetal stem cells to make what appear to be actually relays across the site of the injury with these cells that are derived from embryonic or fetal stem cells. And that appears to improve motor function and sensory function in rats. And so a number of clinical trials are being launched in the next year with different sorts of stem cells for this injury. There's been a refunding of the Geron trial that stopped temporarily for financial reasons. There's a phase 1 safety trial about to start at my place in collaboration with a company called Neuralstem. And there's a similar trial starting up in Switzerland with a company called Stem Cells, Inc. And then we're following these fetal stem cell approaches with approaches based on a similar embryonic stem cell approach to make these progenitor cells that will extend across the site of the lesion in an injured spinal cord. Okay, so let me just close with a remark about both San Diego plus what it's going to take to solve these kinds of medical problems. One of the things about being at Harvard University that I didn't care for was that it tended to be fairly isolated. Different departments were like silos at a farm or at a, I don't know, grain manufacturing plant or whatever. So, you know, I would live in the department of corn, and I would rarely, if ever, interact with the department of soybeans or granola or whatever was hanging around. And so this was not adequate in a way to solve many of the problems that we now tackle. It turns out that medical problems in disease are hard. They're not easy problems. And they actually need teams of scientists to work together with adequate and appropriate financing from an important part of the team, which is the federal or state government, because ultimately you need to build bridges, as we like to do in San Diego, between investigators, with funding partners, with venture capitalists, whoever, that it's going to take to solve these problems in disease. Stem cells are an incredibly important part of the solution to these problems moving forward, but we can't do it without adequate financing, not just of stem cell science in the United States, but all areas of medical science and technology, because stem cells is not a field isolated from all others. It depends upon advances in physical sciences. It depends on advances in genomic sciences. That's part of what we're doing with Greg Venter. And it depends on advances in basic cell biology and our ability to understand how to analyze the behavior of brain cells and other cells in a dish in the cell culture versions of disease that we're setting up and the cells that we're transplanting to try to treat disease. So let me stop there. Thank you for your attention. And I suspect some of you will have some questions for me. Yes, please.
With the ALS, you're talking about using neuronal stem cells to help with the spinal cord stem cells. You said embryonic.
Yeah.
But what is the difference between that and using adult?
So for ALS, there is, in my opinion, no appropriate adult stem cell that can be transplanted into the spinal cord with any reasonable hope of supporting motor neurons and keeping them from dying. The two approaches that are underway that I think have the best promise, one is spinal cord stem cells derived from fetal material, postmortem. That's a stem cell trial that was started by a company called Neuralstem at Emory University, where phase 1 safety trials have been completed and are now moving to phase 2 efficacy trials. And then the second approach that I think will be better, but we have to do the experiments to find out, we don't know until you do the experiments, is with embryonic stem cells being used to make so-called neuronal stem cells in a dish that we then transplant into ALS patients, as we hope to be doing in the next few years. There have been claims that you could use cord blood stem cells. There was a company in Louisiana doing this. That company, I believe, was ultimately shut down because of improper record keeping. And then there's a company in Israel that is doing a trial with so-called mesenchymal stem cells, but where I don't think, based on the literature and based on my analysis of the scientific data, I think it's very unlikely that's going to work. And then certainly all these companies that are taking cells out of fat and shooting them back into the bloodstream, that's just snake oil in my point of view. I don't even think they're stem cells. Not even clear what they're growing or harvesting in this case. Yes, please.
Just a science question here. Do stem cells have a circadian clock? And then my second question here is, when stem cells are differentiating themselves to become other cells, or, you know, like a better word, reproducing, are there circadian clocks? Is it done with the circadian clock perfectly timed as it proceeds in the process?
So let me simplify it to just one question. Do stem cells have circadian rhythms the way we as organisms do, where we sleep at night and become very active in the day? I think the answer to that is I don't know. I'm not sure that that's well worked out yet at all. What I do know is that as a result of work of Steve Kay and many others in the past, there's strong evidence that pretty much every kind of cell has its own cell-intrinsic circadian rhythm and responds to the circadian biochemical clock. Now, whether that affects differentiation of the stem cells into more specialized cells or not, I don't know. I'm not sure anybody knows, or I think nobody knows. It wouldn't surprise me if it had an impact. It's an interesting question.
So no one really knows, like in the making stuff, in the 60th hour, if it's more likely to...
To my knowledge, those experiments have not been done yet in a careful way. They require you, in essence, to watch cells every hour of the day. So there are technical issues with doing them. They're solvable, but again, I suspect that at this point in time, it hasn't yet been the most urgent question, but I'd be shocked if it turns out there's not an impact based on what I know about cell physiology. Yes, please.
Are there any issues with the immune system rejection of stem cells after they're produced?
So the question is, are there issues of immune rejection with stem cells? And the answer to that is absolutely, and there are issues that have to be solved as part of using any stem cell to treat any kind of disease. Best example is when one uses stem cells from bone marrow, which are blood-forming stem cells. If I transplant my bone marrow into the questioner, his immune system is going to recognize those cells as foreign and attack them. And ordinarily, we would use a combination of genetic matching and immune suppression, both for bone marrow stem cell transplant. And of course, we currently use it for heart, lung, kidney, all these other kinds of transplant. The hope is that one day we will either demonstrate that reprogrammed stem cells are usable for these types of disease treatment, and that we get the safety issues under control. Japan is making great progress in that area, and we're all watching to see what happens. It could turn out to be terrific. But also, in the meantime, it's not entirely clear whether reprogrammed stem cells, even if they're made from me, are absolutely identical in the kinds of molecules they express on their surface that the immune system might recognize as foreign, even though they come from me. That's been an area of some debate, and it's still being settled out. By the way, the other approach to it is the approach of a company called ViaCyte, where if cells are making a small molecule like insulin, they're putting the cells in a bag so that they're protected from the immune system as near as we can tell. And then they secrete insulin and it goes outside the bag and into the bloodstream. So that's the other approach that'll work for some settings, is to put the stem cells in a device that protects them from the immune system. And there's quite a bit of work going on in various fields in that way. Yes, please.
Yeah, you touched on the fact that embryonic stem cell research is a politically controversial subject. My question is, for researchers who are applying for grants with NIH for their projects, what kind of roadblock does that?
Yeah, so the question is, are there continuing political roadblocks or regulatory roadblocks to using embryonic stem cells with funding from the NIH? So there's sort of two answers to this question. So first of all, I love living in California. And those of you who are not from California, sorry, I just can't help it. I'm a Californian. And of course, we have a funding agency that helps us with some of those problems. I think the second answer, though, is that we will never be free entirely from any sort of regulatory burden using human embryonic stem cells. And actually, that's as it should be. We should regulate the area carefully. We should be sure that appropriate informed consent is used when cells are donated, and that researchers such as myself follow the rules when we're using these cells and have appropriate and justifiable scientific or medical projects using them. Now, I'm going to argue that Bush got a bad name on this issue in the following sense. When Bush first opened the door to federal funding of human embryonic stem cell research, A, it was the first time that federal funds could be used with these types of stem cells, and B, at the time, it was all available stem cell lines, so-called. Since that time, there's been a change from the Bush policy to the Obama policy, because the Bush policy aged poorly, because newer stem cell lines that were made, for example, in the Melton Lab at Harvard and elsewhere, were ineligible for federal funding because the lines were generated from embryos that were destroyed after the date of President Bush's speech in 2001. Now then, just because those stem cells are now usable under an updated Obama policy, there are still some very strong ethical guidelines about how they can be used. And somewhat surprisingly, there are stem cells that even under the Obama policy cannot be used freely because of inadvertent restrictions in the informed consent documents. And so we're still having to work around some of these restrictions, and we still track things pretty carefully as a result. Now, that could all be jeopardized by any new administration or Congress that comes in and wants to change the rules. And so, you know, we in California, where we have some protection and constancy in this area, because even though it's controversial, we wrote some of this into our state constitution to protect this area of research, that could change federally. And so I don't think we ever want to lose sight of the fact that this is valuable research using embryonic stem cells. The federal government is an important player, and it should remain an important player if it wants a strong national effort. Yes, please.
How are you able to take a specified human cell and knock it down to becoming a stem cell? Do you just knock out a couple of genetic sequences? Or?
Okay, so I think the question is, what is this mysterious reprogramming technology that I mentioned? And I sometimes call it genetic trickery. So here's what's done in essence, and I'll try to avoid the technical jargon. Embryonic stem cells activate certain genes that tell them to be embryonic. And those genes tell them divide and grow well in the lab, amazingly, and retain the ability of all early embryonic cells to make all adult tissues. When you take an adult skin cell, those embryonic genetic elements are turned off for the most part, and different parts of the DNA are activated, different genes are activated, and those genes say, be a skin cell. Make skin proteins, such as keratin, don't divide very often, and when you do divide, only do it when you get an instruction from cells in the skin called the niche, the environment those cells live in. What we do in terms of the genetic trickery is we can isolate some of those genetic elements from the embryonic cells. We can put them into viruses or other carriers that allow us to introduce those embryonic genetic elements into an adult cell. And they will override the genetic activity in those adult cells and convert them into an embryonic-like cell. Here's the example I give to help you think about that. You guys know about singing in the round, right? So the idea is, suppose you have a choir and it's singing verse A of some song. Everybody's singing in tune, they're singing along together, and then one person starts singing a different song. And then that spreads throughout the choir, so that now everybody is singing song B. That's in essence what we're doing, is we're implanting those genetic instructions, activating them, and fooling the cell into becoming a reprogrammed stem cell. Amazing technology. We all thought it couldn't be done. They're all wrong. It happens. We get over it. Yes, please.
If I understood you right, you were saying there was some sort of bond initiative for embryonic stem cell. I was wondering if you could describe that and compare maybe how things are going in California versus other states.
Sure. So the question is, what the heck is going on in California with stem cells? Wasn't there some mysterious bond initiative? So Bob Klein, who has a son with juvenile diabetes, type 1 diabetes, and a number of patient advocates and scientists got together and wrote a bond initiative in California. California, you may recall, is kind of unusual in American states because it allows direct governance from the people, the voters, under some circumstances. And so in California, a bond initiative was written to borrow $3 billion to fund stem cell research over 10 years, about $300 million a year. That bond initiative got on the ballot, and then 60% of the people of California voted in favor of borrowing money to fund stem cell research in the long term. Now, as a result, there are a number of stem cell projects in California, ranging from the very basic science of stem cells and different cells you can make from stem cells, right through to clinical trials shortly of stem cell-derived products or drugs targeting stem cells in cancer or stem cells for ALS or what have you. That's after 7 years of financing. As I understand it, the numbers are we've spent about $1 billion of state money. We've attracted $1 billion or more in additional capital into the state that we've leveraged off of that state money, and we are just getting started. Watch out. Sorry, I'm a Californian, as I said. Yes, please.
Earlier you mentioned that stem cells have a promise in drug development. I was wondering if you could elaborate on what kind of potential stem cells have for the development and expansion of biologic and biosimilar medications.
Sure. So the question is, stem cells seem to be useful for finding and testing drugs. What about other types of biological agents such as antibodies or growth factors or cytokine antagonists, such as I take actually, or other types of biosimilar molecules? And I think my answer would be everywhere where you can do drug testing for stem cells, if there are clues about biologically based agents that you might trust, that you might be interested in, you can set up systems to test them in stem cell derived materials in a dish. Now, that's a little more complicated for cells of the immune system, where we're not very good yet at making them in a dish from either bone marrow stem cells or embryonic or reprogrammed. But that's a technology that appears to be rapidly improving. And of course, if anybody had some good ideas about biological agents for Alzheimer's disease, I'm pretty sure that we could think of a way to develop a system for testing them. We're just not there yet in many ways, but it's coming. Yes, please.
With the use of stem cells nowadays, especially the embryonic stem cells, there's always the downside of the formation of teratomas. I'm just wondering what biomedical technologies have been developed to overcome this problem.
Yeah. So the question is, with embryonic stem cells and reprogrammed stem cells, and by the way, probably every type of stem cell that you transplant or cell type that you transplant from one person to another, you worry about tumor formation. You worry about it to different degrees depending on the stem cell source. With embryonic stem cells and reprogrammed stem cells specifically, if you take one of those cells or a group of those cells and transplant them directly into an animal where the immune system is shut down, they will make a non-malignant kind of tumor called a teratoma. That's just fact. They will make that. And so the concern is, if I take my embryonic stem cells and I induce them to form so-called neuronal stem cells, am I worried about those neuronal stem cells forming teratomas when I transplant them into a rat and then into a human? And the answer is, of course I worry about that. And what we do is we test to see whether that's happening or not. So let me just tell you two pieces of data about that that have developed over the past 10 years. A, when we grow neuronal stem cells in the lab, they grow in an entirely different type of growth medium than that which we use for embryonic stem cells. That's part of how we induce them to so-called differentiate, to convert from one type to another. Embryonic stem cells, even in the optimal growth medium, are what we call, is an important technical term, cranky. They're cranky in the sense that they're very finicky about the medium and how you treat them and when you feed them and what temperature. They're actually hard to keep growing well in the lab. And so if you induce them to convert to another type of stem cell entirely with a completely different type of growth medium, they don't actually contaminate those cells very much, as near as we can tell, because we've put them in hostile conditions. Now, that's an experimental statement. That's what we've observed. But then second, we can take those neuronal stem cells, we can implant them into rats or mice and test them directly for tumor formation. And the answer is, using the types of stem cells that we're using thus far, we have seen no evidence of tumor formation. So if the stem cells that you're making are well prepared, or the so-called terminally differentiated cells, such as those being tested in the eye for macular degeneration and other diseases of the eye, if they're well differentiated, carefully tested, there appears to be virtually no danger of teratoma formation. You always have to test. The FDA requires us to do a lot of safety testing, perhaps too much in the eyes of some, perhaps not enough in the eyes of others, but we absolutely do test it, and that's an important part of our development program, and that's part of what takes time and money. Yes, please.
How can stem cells be used to understand the effects of environmental exposures on people?
Yeah. The question is, can we, and if so, how do we use these kinds of stem cell-based technologies to understand environmental toxins? Because there are a lot of them out there, you may know, and they're rather dangerous in many cases, such as rotenone we were talking about earlier, that increases the risk of Parkinson's disease, but it's an ingredient in many common pesticides. So here's the issue. Rats and mice are not just little humans, and we are not just big rats and mice, right? So chemicals that are toxic to a human are not necessarily toxic in the same way to rats or mice, and by the way, vice versa. And many of the proteins and enzymes in the liver that are ordinarily involved in the breakdown of environmental toxins, they're not the same in humans as they are in rats and mice. And in fact, there's a huge amount of genetic variation in those systems in humans, so that any one of us will respond slightly different to different sorts of toxins. Some of us will be very sensitive to one kind of toxin, and others of us not at all. So here's how this might be useful or usefully tackled with stem cell technology. Suppose I make stem cell lines from everybody in this room the way I did from Craig Venter. And I test how each of you respond to an environmental toxin, not you the person, but you, your stem cells that I say make into liver cells in a dish, which is a common source or source of cells that detoxify environmental toxins. I can then ask, how do each of us respond? I can find those that are, for example, super sensitive or super resistant, and then we have tricks for tracking down which part of the DNA is responsible for sensitivity or risk, and then we have tricks for introducing, say, the risk variants into a resistant cell line to be sure that we've actually identified the right risk variant by introducing them with genetic engineering technology and then making, say, liver cells in a dish. This is all just getting going, but it's a fabulously important use of this technology, not just for environmental toxins, by the way, but we know whenever we develop a new drug, we do a clinical trial. We do a clinical trial, and that tells us how the average patient responds to a drug. And I can just tell you, as someone who has a chronic disease, there are plenty of drugs that have been through clinical trials that don't work on me, or even make me sick. And then there are other drugs that work great on me, but don't work on somebody else. Now, that's something that you really hate discovering after a drug has been on the market for a while, but that's how it's currently done. If we can use, as we ultimately hope we can, it's not going to be this year or next year, it's going to take a while to develop this technology. But if we could have used, say, my stem cells to make the appropriate cell type from me to test it for response or not to those drugs, it would have saved me several months of being sick when we were trying out a new drug on me. And that, to me, is a really great use of this technology. Well, if there's nothing else, thank you for your time, folks. I really appreciate it.